SNEV Translational Series, and I would like to thank you for joining us. My name is Débora Ferreira, and I'm a postdoc at Portugal. Today I have the pleasure of moderating this session alongside my colleague, Helena, that is a PhD student from the University of Porto, also in Portugal. It is a great honor to introduce you our special guest, Professor Francesca Fallarino. She is a full professor at the University of Perugia, where she teaches immunopharmacology with a focus on advanced therapies. Her research explores how dendritic cells and macrophages can be trained to overcome tumor immune resistance or modulate autoimmune responses. Recently, our group discovered that natural extracellular vesicles and dietary components can influence the dendritic cell function by activating specific metabolic sensors. Her work has indeed advanced the understanding of the link between the metabolism and the immune regulation in myeloid cells, paving the way for next-generation immunotherapies that bridge the basic science and clinical application. I kindly ask to the audience to submit their questions in the chat box. These will be addressed in the end of the session. And Dr. Francesca Fallarino, thank you once again for accepting our invitation. We are thrilled about your talk and excited to hear your insights. You may now begin your presentation. Thank you.
Thank you very much for this kind introduction, and also for the opportunity to share our recent findings in the area, in the field of extracellular vesicles that indeed are in fields or of really exciting finding in physiology and also in pathology. So I would like also to take the opportunity to introduce the extracellular vesicle network at the University of Perugia. This network brings together several researchers and faculties at the University of Perugia that are sharing their technologies and also their expertise. And in particular, through this network, we can share expertise in EV isolation, EV characterization, and also in the ability of EV to modulate specific biological functions. And also we have expertise in bioinformatic analysis of the cargo of these extracellular vesicles. So we are actually open to national and international collaboration, because we think that to advance this field, it's important to exploit collaboration among different researchers. And so here you also can find the specific email that you can also contact us if you want to have some information. So before we dive into the findings of research findings that involve extracellular vesicle, I would like also to introduce the agenda of this today's presentation. So we will start with a brief introduction about extracellular vesicle, covering mainly their characteristics and also their biogenesis and biological functions. And then I will share our recent findings, mostly in the characterization of one type of extracellular vesicle, mostly the amniotic extracellular vesicle, and highlighting their immunoregulatory function or to regulate myeloid cells. And then we will make some conclusions and potentially some discussions. So let's start with the first part. So what are indeed extracellular vesicles? So the extracellular vesicle, this is a term that was proposed in 2011, and we just mean intact lipid bilayer vesicular structure that are released by all body cells, and they are present in all body fluids like milk, blood, synovial fluid, and also, as you will see in the presentation, in amniotic fluid. So they can carry and transport several bioactive molecules, such as protein, but also nucleic acid, like messenger RNA, microRNA, DNA, and also metabolite. And for this reason, they are very important because they actually reflect the makeup of the cell that produce the extracellular vesicle. So there are different types of extracellular vesicle that we can distinguish also by the secretion pathway. So the exosome, for example, are produced by and are formed by the multivesicular bodies. And in general, their size, it's between 30 to 150 nanometer. And then we have the ectosome or microvesicle, and their size is between 100 and 1000 nanometer. And then also we have apoptotic bodies that are delivered during cell death. And so if we think that all cell types in the body are producing extracellular vesicles, we have trillions of these vesicles that are circulating in our body. So how these extracellular vesicles are originated? So there is not really a specific extracellular vesicle pathway that we can actually distinguish, but there are some major pathways like the ESCRT dependent pathway that are very important for the production of exosome-like extracellular vesicle, but also other ESCRT independent pathway that involved the lipid rearrangement and also cytoskeleton rearrangement and the production of other type of extracellular vesicle. So because of this biogenesis, extracellular vesicles express on the cell membrane, and you can see here in the right left hand side of the slide, some proteins that are involved in transport, extracellular vesicle transport, and also membrane fusions, like integrins or tetraspanins, and also there are molecules that are involved in immune regulation, as we will see during the talk. So for what I said till now, so extracellular vesicles are produced by a donor cell, and this donor cell release extracellular vesicle close by the cell and mostly in the extracellular space. And these extracellular vesicles can act both in the paracrine and endocrine function. And so they can act as a reservoir of several metabolites or molecules in the extracellular space, but also can be taken up by recipient cell. And so this recipient cell, upon contact and delivery of this extracellular vesicle, they can change their function. They can change their gene expression profile, but also they can change signaling. And so how this happens? So we can subdivide this uptake by three different major mechanisms. So the first one is the membrane fusion. And the second one is cluttering-dependent endocytosis. And the third one can be a cluttering-independent endocytosis involving phagocytosis, pinocytosis, and also the formation of lipid rafts and protein-protein interaction have been also suggested for uptake of extracellular vesicle by recipient cells. So, but when an extracellular vesicle interact within a specific ecosystem or with specific cells, what are the effects? So what are the functions? So originally, the extracellular vesicle have been thought to be a way to dispose some waste by the cell, but actually the functions are more diverse than that. And so the extracellular vesicle can circulate into vessels, and they can also interact with extracellular matrix and can interact, as we said, with specific cell type, regulating their function. And so for this reason, extracellular vesicles have been proposed to modulate several biological functions that go from immune modulation, tissue regeneration, and also delivery of specific bioactive molecules. They are very important also and consider important tools as a biomarker because they reflect the makeup of the donor cell and can be very important as well as a diagnostic tool because they can carry bioactive molecules from different cell types. So just summarizing all these characteristics, we can assimilate the extracellular vesicle as a sort of message, a reservoir of bioactive molecule that can be important means to maintain a network within cells, tissues, and organs. So similarly to what happened to the social network with the different tools that we use every day to interact with different people. So regarding the second part about what we know about the extracellular vesicles, as I said, the extracellular vesicles are enriched in several body fluids, spanning from saliva, milk, blood, and urine. And some years ago, in collaboration with a colleague at the University of Perugia, Rita Romani, we identified specific stem cells that we could isolate from human amniotic fluid that we were taken from the first trimester during pregnancy, during normal analysis of pregnant women. And so these amniotic fluid stem cells had peculiar characteristics, as I will show you in a second. But very recently, we found that these cells are capable of producing a large amount of extracellular vesicles. And we called these extracellular vesicles, for this reason, human amniotic fluid stem cell derived extracellular vesicle. More recently, we also found that within the amniotic fluid, there are heterogeneous extracellular vesicles that we have called human amniotic fluid extracellular vesicle. So why we started to explore these extracellular vesicles from the stem cell from amniotic fluid? So we need to go back a little bit in our study, and we found that this stem cell had the characteristics to be a pluripotent stem cell, and they were capable of self-renewal. They could differentiate toward osteogenic and adipogenic phenotype. But when also we were co-culturing this specific stem cell with PBMC, we could find that these cells were inducing immune modulation, meaning they were suppressing T cell activation and they were promoting the differentiation of FOXP3 positive T cells, like Treg cells. And so what was really intriguing was the fact that this effect could happen both in cell-cell contact system, but also in transwell system. So did not require direct cell-to-cell contact. And so this suggested that something that was in the cell supernet and was released by the stem cell could modulate immune responses. So we tried different molecules, but a single molecule was not able to reproduce such effects. So more recently, in collaboration with another colleague, Luisa Pascucci at the University of Perugia, we actually could demonstrate that this specific stem cell isolated from the amniotic fluid were capable of producing extracellular vesicle. Here you can find by this electron microscopy that these cells were capable of producing ectosomes and also forming multivesicular bodies. So this suggested that these specific stem cells were capable of producing both ectosome-like and also exosome-like extracellular vesicles. So just strong from this finding, we set a specific isolation workflow to isolate these specific extracellular vesicles. So we culture the stem cell in specific serum-free media, and then by differential centrifugation, we isolated the extracellular vesicle. And I will focus in this talk mainly on the extracellular vesicle resembling exosome-like EVs. So these EVs were then deeply characterized by sites and also by morphology, and also we characterize for their function. So here I will show the characterization that we achieved. So by the scanning electron microscopy, we could demonstrate typical spherical shape of this vesicle and their specific the specific sites around 80 nanometer. And also by using the nanotracking analysis, we could actually show similar sites, as you can see here. And by Western blot analysis, we could also demonstrate the presence of markers typically expressed by exosomes, like helix, TCG101, and also ARF6 and CD81. So then we also next analyze, we wanted to analyze more deeply this extracellular vesicle. And first we analyze the lipids content. And so we compare the lipid content also with the donor extracellular, the donor cell that produce extracellular vesicles. And as you can see here, there were several lipids that were overlapping between the extracellular vesicles and the donor cells. But there were some lipids that were more enriched in the extracellular vesicles, such as, for example, the sphingomyelin and also the lysophosphatidylcholine, suggesting that these specific lipids could be important for the uptake or maybe the delivery of these extracellular vesicles. We also performed proteomic analysis, and we found around 47 major protein that were enriched in these extracellular vesicles. And among these 47 protein, 44 were already found in the Vesiclopedia, so they were overlapping already with the described protein, but we found also 3 additional new ones And we are trying to explore these new one that are typically expressed by these extracellular vesicles. The protein were involved in amniotic fluid and were found already in amniotic fluid and also in other body fluid like saliva or urine. We perform also transcriptomic analysis, and we identified by the reactome system several messenger RNA that were involved in immune regulation, so in complement activation, in immune response of lymphocytes and inflammation in different organs. Also, we identified more than 300 microRNA that were expressed by these specific extracellular vesicles, and several of them were involved actually in immune regulation. All these results then suggested that in the original culture that we saw regulation by amniotic fluid stem cells, maybe extracellular vesicles were playing a very important role. And so next, to try to see whether these extracellular vesicles were taken up by immune cells, we actually stain extracellular vesicle with a specific dye, and then we put in culture this extracellular vesicle with either murine splenocytes or human PBMCs. And then we recorded the DIL positive in this culture, positive cells in this culture. As you can see, in this culture, we found several cells that express this specific dye. And so meaning that different cells were capable of interacting with extracellular vesicle, both in the splenocytes and from mouse models and also in human PBMCs. So by using specific antibodies, recognize specific cell types, we also found that these extracellular vesicles were mainly interacting with cells characterized by myeloid origin, such as macrophages or subsets of dendritic cells, both in the murine model and in the human system. So to confirm this data, we also injected this stained extracellular vesicle or vehicle containing the same dye in vivo, and then we recorded the dye positive cells also co-expressing specific markers, and so we could confirm in the splenocytes that indeed these extracellular vesicles were taken up by macrophages mostly and dendritic cells. And among dendritic cells, the cells that were more capable of interacting with this extracellular vesicle were a type of dendritic cell, the conventional dendritic cell type 2. So we can imagine in vivo that several extracellular vesicles are produced by different cells, and so in any time, so there is probably an equilibrium of these different extracellular vesicles. So if we have an equal amount of extracellular vesicles that are producing pro-inflammatory and anti-inflammatory function, so we don't see any effect. But if we have some extracellular vesicles that are pro-inflammatory, so we can stimulate immune response, and then if we have extracellular vesicle with anti-inflammatory function, we have a suppression of immune response. So we were really interested to see which effect these extracellular vesicles could induce in dendritic cells. But first, we wanted to see whether these extracellular vesicles could be taken up or just interact with dendritic cells and macrophages. So previously, in collaboration with Letizia Mezzasoma also at the University of Perugia, we showed in monocyte cell line THP1 that the addition of extracellular vesicles from human amniotic fluid stem cells was able to suppress inflammasome activation, leading to suppression of caspase-1 and also decreased production of cytokine like IL-1 beta and IL-18. So dendritic cells are very important cells in regulating immune responses because they can regulate both innate but also adaptive immune responses. And also, dendritic cells are very plastic cells, and so they can be highly influenced by environmental function and environmental signals to induce immunity and tolerance. And so, but the dendritic cell landscape is very complex, and dendritic cell can be simply subdivided into three major subtypes: the plasmacytoid, the conventional dendritic cells, and the monocyte-derived dendritic cells. I will show you the results on the conventional dendritic cells and mostly on the CDC2 and CDC1, just because we found that extracellular vesicles mostly interact with this type of dendritic cell. So we asked whether these extracellular vesicles from human amniotic fluid stem cells were capable of regulating inflammatory DC and leading to regulatory CDC. In previous findings from our lab, we demonstrated that indeed CDC1 and CDC2 responded differently to an inflammatory signal. As you can see here in this graph, CDC2 responded in a very fast way to stimulation to LPS and leading to production of pro-inflammatory cytokines like IL-6, while CDC1 responded more slowly, and also they did not reach this high amount of IL-6 production. Actually, we found that CDC1 had a more regulatory phenotype than CDC2. And so we wanted to actually address whether extracellular vesicles from human amniotic fluid stem cells were capable of interacting differently between CDC1 and CDC2. And as you can see here, we found that these extracellular vesicles were capable of interacting with both CDC1 and CDC2, but while confocal microscopy study revealed that in CDC1, the extracellular vesicle remained on the periphery of the cell, so on the cell membrane, while in CDC2, the extracellular vesicles were internalized. And so this suggested that these extracellular vesicles were capable potentially to modulate strongly the CDC2 phenotype. And here you can see the results with the confocal microscopy that are just demonstrating what I said. So we were particularly interested to try to address how the CDC2 could actually internalize and uptake these extracellular vesicles. And it has been reported that integrins are very important for cell uptake of extracellular vesicles, and in particular, integrin beta-1 and integrin beta-3. And so we looked at the expression of these two integrins in different immune cell types. And we found that both in the murine model and in human PBMC, there was a higher expression of integrin beta-1 compared to integrin beta-3. And so we hypothesized that this integrin could be involved in uptake of extracellular vesicles by the CDC2. So by using antibodies blocking this integrin, also called CD29, we could demonstrate that there was a significant reduction of extracellular vesicle uptake by murine CDC2 and also by human CDC2. So this suggested that this integrin is important for interaction of this specific extracellular vesicle with the CDC2 cells. But this interaction is able also to activate a specific function on CDC2. And so in order to do that, we first incubated CDC2 with extracellular vesicle from human amniotic fluid stem cells, and then these cells were treated with LPS. Here you can see nicely how these extracellular vesicles are interacting with the CDC2. And then these CDC2 were also analyzed for the ability to produce pro-inflammatory and anti-inflammatory molecules. So you can see that this treatment reduced significantly the production of pro-inflammatory cytokines like IL-6, IL-12, IL-23, TNF-alpha, and also increased anti-inflammatory molecules like LAP expression on these cells. We also found that the increased amount of extracellular vesicles were also involved in the reduction of MHC class II and therefore potentially influencing antigen presentation on the CDC2. But this specific effect on mostly innate response has also some effects on adaptive immunity. In order to address this second function, we also took CDC2 that were preconditioned with extracellular vesicles from amniotic fluid stem cells, and then we co-cultured these dendritic cells in the presence of the OVA antigen with transgenic T cells that respond specifically to the OVA antigen. And then we address T-cell activation, T-cell proliferation, and T-cell differentiation in vitro. We found that this specific conditioning of the CDC2 significantly impacted the T-cell activation by reduction of the expression of the CD44 by the T cells, and also impact on T-cell proliferation, as you can see here, by reduction of T-cell proliferation to increase concentration of the OVA antigen. And also this precondition also increased the differentiation of T cells toward Treg expression and differentiation of FOXP3 positive T cells. So all these results actually suggested that preconditioning of CDC2 with extracellular vesicle derived from this specific body fluid was capable of regulating in a negative way immune responses. So dendritic cells have been strongly involved in induction of autoimmune response, and specifically in multiple sclerosis. In fact, they can present self-antigens and they can prime T cells that also can infiltrate the CNS and then also react against myelin antigen. And these T cells mostly express pathogenic cytokines like IL-17 and interferon gamma. So to mimic this specific disease, it's possible to use a specific preclinical model that is called EAE for experimental autoimmune encephalomyelitis. And so in this model also it has been suggested that the presence or the absence of specific dendritic cell subsets can strongly influence the disease onset and severity. And in particular, also our findings, we demonstrated previously that the absence of inflammatory CDC2 actually is able to completely prevent this disease in this mouse model, while the depletion of the other subset, the CDC1, actually increase EE severity. So this suggests that indeed CDC2 are the cells that are involved in the pathogenesis of this specific disease. So it is important to regulate such dendritic cells in order to decrease the severity of the disease. And so in order to actually exploit this hypothesis, we perform this specific in vivo model where actually we induced EAE in vivo by immunizing the mice with the specific myelin MOG peptide. And then we boost the immune response with the adjuvant pertussin toxin. And then we treated the mice for 2 times with CDC2 either not conditioned or conditioned with extracellular vesicles derived from human amniotic fluid stem cells. And then we recorded the severity and the score of the disease, as well as immune infiltration in the CNS and cytokine production. Here you can see the results. So you can see that the conditioning of the CDC2 and the administration of these specific dendritic cells conditioned with the extracellular vesicle significantly reduced the score and the severity of the disease in this specific model, while CDC2 alone, unconditioned or vehicle, did not have any effect. What was also very interesting was the fact that in these mice, the administration of CDC2 conditioned with extracellular vesicle led to myelin integrity, more myelin integrity in these mice, and also reduce immune infiltration. We characterized very deeply also the immune infiltration using multiparametric analysis and flow cytometry analysis, and we found a different landscape of the immune infiltration in mice that were treated with CDC2 treated with vehicle or CDC2 treated with extracellular vesicles. And by analyzing the production of pathogenic cytokines produced by immune cells taken from these mice and from draining lymph nodes, we found that indeed administration of these dendritic cells conditioned with extracellular vesicle led to the reduction of pathogenic cytokine production like IL-17A, interferon gamma, and led to the increase of anti-inflammatory cytokine like TGF-beta and IL-10. And also similarly to what we had found in vitro, there was also an increase of FOXP3 mRNA expression and Treg differentiation in these specific mice. So as a summary of these specific results that I showed you till now, we found that from human amniotic fluid, we can isolate specific stem cells that produce large amount of extracellular vesicles, and these extracellular vesicles can modulate the function of professional antigen-presenting cells, such as CDC2 cells that are inflammatory in specific contexts. And also this interaction led to the modulation of CDC2, and that can acquire tolerogenic phenotype, and this tolerogenic phenotype can promote Tregs and protect against specific autoimmune diseases such as EAE. So just to conclude, I want just to show just a brief result about the other type of extracellular vesicle that are indeed isolated and can be indeed isolated directly from human amniotic fluid. So these extracellular vesicles are more heterogeneous, and we found some time ago that these cells can either trigger or inhibiting inflammasome activation. In fact, we found that this extracellular vesicle contained a specific endotoxin. So this suggested that maybe they were also carrying specific microbes. And here it's really a debate about how sterile can be the amniotic fluid. So by doing all the controls and doing metagenomic analysis, we found indeed that in the extracellular vesicles enriched directly from amniotic fluid, we could find traces of microbiota species variants, and these were absent in control environmental samples. So this actually led to the actual hypothesis that we want to exploit, that these specific microbiota species can maybe train the fetus early on to be tolerant, for example, to specific microbiota species that will colonize also the gut. So with this last result, I want just to thank the people that actually collaborated and did the experiment. And in particular, I presented the data that were actually performed by postdocs and researchers in the lab, Giorgia Manni, I mentioned already Rita Romani, Marco Gargaro, Doriana Ricciuti, Letizia Mezzasoma, Estevao Barcelos, and also for the macrobiota, Teresa Zelante. I want also to thank the collaboration within the Extracellular Vesicle Network at the University of Perugia, and also the long collaboration with Professor Kenneth Murphy that provide a very important tools to exploit dendritic cell function. And I would like also to thank the Immunometabolism and Reactivity Lab at people at the University of Perugia, and all of you for the attention. Thank you very much.
Thank you so much, Dr. Francesca, for your insightful talk. The session is now open to questions. You can either unmute yourself or put your questions in the chat box.
Perché lei sta facendo la videoconferenza europea?
Ah, certo, è stata portata questo...
Rita, do you want to make a question? No. We have a question in the chat box from Débora. I don't know if you want to unmute yourself, Débora, and ask yourself.
Really nice work. Thank you for sharing with us. My question is about your EV lipidomics, transcriptomics, and proteomics. If you try to integrate the data in order to find an, I don't know, maybe a phenotype or something.
So this is an interesting question, and we have been trying to integrate the data from lipidomics, transcriptomics, and also proteomics. And we have several overlapping molecule between proteomics and transcriptomics. And also we have microRNA that are also regulating the expression of specific molecules. So this it's a sort of network of biological molecules that indeed can regulate each other. And so we think that all together actually give the result that I showed you, because it's a balance for sure.
Thank you.
We have another question in the chat from Linoy Israel. Do you want to unmute yourself, or maybe I can read it? So she wanted to ask, how did you separate the specific EVs from all the fluid?
So we separated the EV by differential ultracentrifugation, so at different speed. So we took the supernatant and then we subjected to ultracentrifugation at different speed. So to isolate first the microvesicles and the exosome-like. And also recently, we are doing also columns to exclude the protein. So to have more actually purer extracellular vesicles and eliminating contaminating proteins.
Thank you. We have another question in the chat from Julio, who asks which immunogenic determinants were characterized in the amniotic fluid derived EVs.
Immunogenic. So mostly we found actually in the amniotic fluid, so the other one. So mostly we found traces of ligands that are capable of activating TLR ligands. So it seems like toll receptor ligand that were isolated in this extracellular vesicle from amniotic fluid. And so, in fact, when in this study that we have published, we found that if we treat macrophages with this extracellular vesicle one time, we have actually production of pro-inflammatory mediator. But if we treat twice in sequence, we have actually a reduction. And this is similar to what happened to LPS tolerance. So this means that there are molecules that can first stimulate, and then if we use as a second time, can suppress immune responses. And indeed, we found endotoxin-like molecule in this extracellular vesicle that were not contaminant from the environment. So because we did all types of controls, so it's just in there. And we also isolate this amniotic fluid extracellular vesicle in very controlled situation and conditions, and always these molecules were there. And so for this reason, this prompted us to analyze, to do and perform metagenomic analysis.
Thank you so much. So also in the chat, Natasha Oliveira is asking, have you tested other type of stem cells source to confirm if this result is specific of amniotic fluid stem cells or can be extended for other cell types?
Mm-hmm. Yes, it's very interesting also this question. And so we are actually thinking to perform and to analyze also the effect of other stem cells, specifically to in the effect on dendritic cell subsets, because this is quite new. Instead, for macrophages or dendritic cell in general, there are already results in the literature that this extracellular vesicle can regulate the function of this myeloid cell. But there are not so many data in specific dendritic cell subsets. And this will be also the subject of future study.
Thank you. We have another question from Vanya. She asked, under what conditions should the amniotic fluid stem cells be maintained? In my case, why is it that after passing them to the next culture, they begin to differentiate and do not grow well?
Yeah, so this it's quite complicated. I understand it's true. Also, we had a lot of difficulty, and together with the colleague here at the University of Perugia, Professor Rita Romani, actually we established specific culturing conditions, and also we found that in different samples, you don't really have always lines that you can establish long time. So there are two different ones. If you look at our publication in 2015, you can find that there are two types of stem cell from amniotic fluid. One that grows really fast, and we call fast, and other that grows really slow, and we call slow. So it depends. It depends on the donor. So we don't know exactly why you have one or the other. But in general, if you use the condition that we also published, they don't differentiate. So they stop to proliferate, but they don't differentiate. You need to use specific media for stem cell.
Okay, thank you. Maybe I would like to ask regarding the mouse model that you use for the preclinical MS model.
Mm-hmm.
Is it gender specific, only female or male? Can gender, in this case, have an influence on the immune response to these dendritic cells treated with amniotic fluid?
Mm-hmm. So in general, we don't know exactly in the male because everything is female. So the EA model is actually done also in female, but also we found that these immunoregulatory effects of dendritic cells, especially for the subset, can also function in male. So we don't think that is a gender specific effect, can be also both in female and male regulation.
And do you think that we could use these EVs in humans in the future to treat patients? Could we have the resources to do it in the clinics to isolate EVs from the amniotic fluid as a routine process?
Yeah, this is what we are thinking together with the colleagues at the University of Perugia to try to exploit these finding specific settings. And so, because we think that in comparison with the stem cell, could be easier in a way to translate also for treatment. And also we have several stem cell lines that can be used as a source of this extracellular vesicle, or also the extracellular vesicle can be isolated from amniotic fluid. But in order to actually control better all the conditions, would be better to isolate from the stem cell, because we can characterize and we can control all the parameters in a better way. But yes, we are thinking to try to exploit these results also to treat some inflammatory response.
And the amount of EVs that we can isolate from this amniotic fluid, do you think that we could reach the amount that would be needed to inject in a human? Because in comparison with a mouse, I imagine that we would need a much higher quantity of EVs.
Yeah, yes, this is also a critical question. I understand, and this is something that we are discussing. But what actually we found also in vitro study, that we don't need a lot of this EV to regulate. Actually, we saw some effects that are better when we use a low amount of EV compare a large amount of EV. So this suggests that this can be feasible. But yes, we need to think in a very critical way about this issue.
Thank you so much. That's a very great work. I don't know if anybody else would like to make a question. You can also unmute yourself. If no, maybe we could end this session, and thank you so much, Dr. Francesca, for this talk and for answering all our questions. I don't know if Débora would like also to say some words, but
just to thank you your kind ability to be here to answer the questions and to your really nice work to share with us.
Thank you very much to all of you for inviting me to this online talks. Was really enjoyable, and thanks again for the discussion as well. Thank you.
Thank you.
thank you.